Welcome to our exploration of the cell cycle. The cell cycle is a fundamental process that occurs in all living organisms. It describes the complete journey a cell goes through from its formation to the moment it divides to create new cells. Think of it as the life story of a cell from birth to reproduction. So what exactly is the cell cycle? It's a series of organized events that a cell undergoes as it prepares to divide. During this cycle, the cell grows larger, makes a copy of all its DNA, and eventually splits into two identical daughter cells. This process is continuous and repeats throughout the life of an organism, allowing for growth, repair, and reproduction at the cellular level. The cell cycle can be divided into two main phases. First is interphase, which is the growth and preparation period. During interphase, the cell spends most of its time growing and getting ready for division. Second is M phase, also called the mitotic phase, where actual cell division takes place. Think of interphase as the planning and preparation stage, while M phase is the execution stage. Interphase itself is divided into three distinct subphases. The first is G1 phase, where the cell grows and carries out normal functions. Next comes S phase, where DNA synthesis occurs and the genetic material is duplicated. Finally, there's G2 phase, where the cell makes final preparations for division. Each of these subphases has specific tasks that must be completed before the cell can proceed to the next stage. Let's look closer at G1 phase, also called gap 1. During this phase, the cell is actively growing in size and volume. It produces new organelles, which are the tiny organs inside the cell, and synthesizes proteins needed for various cellular functions. The cell also accumulates energy in the form of ATP, which will be needed to power the division process later. This is essentially the cell's growth and strengthening phase. Next comes S phase, where synthesis occurs. The most important event here is DNA replication. The cell makes an exact copy of all its genetic material. Each chromosome, which contains DNA, is duplicated to form two identical copies. This duplication is crucial because when the cell divides, each daughter cell needs to receive a complete set of genetic instructions. Without this step, the new cells wouldn't have all the information they need to function properly. The third subphase is G2, or gap 2. During this phase, the cell continues to grow and begins producing specialized proteins that will be essential for mitosis. These proteins include those that will help separate the chromosomes and divide the cell. G2 is essentially the final checkpoint and preparation phase before the cell commits to division. Once this phase is complete, the cell is ready to enter mitosis. Finally, we reach M phase, where mitosis occurs. This is the dramatic phase where the nucleus divides and the cell physically splits into two daughter cells. The duplicated chromosomes are separated and distributed equally to each new cell. At the end of mitosis, each daughter cell receives an identical and complete copy of the DNA. These two new cells are genetically identical to each other and to the original parent cell, and they can now begin their own cell cycles.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.